I worked on an RNA decay pathway known as nonsense-mediated RNA decay, and in short, I'll be calling it NMD. NMD degrades nonsense mRNAs, which have premature translation termination codons and prevent expression of truncated proteins which might be lethal to the cell. When I first started this project, there was no other study that had been done to fully characterize the factors that are important for NMD in mammalian systems. And the genes that we knew were important for NMD were identified in yeast and C. elegans. And so it was important to use an unbiased way to identify genes that were important for NMD in human cells. So the first thing that we did was develop an NMD reporter system that can accurately and efficiently measure NMD in individual cells. So we made a reporter with 2 transcription units, one containing mCherry and a premature termination codon, and one containing a wild-type gene and GFP as a control. So in this way, we can measure NMD as a ratio of mCherry containing protein to the GFP containing protein. And we were able to separate cell populations based on NMD activity, looking at the mCherry containing protein versus the GFP containing protein. So with this reporter, we were able to perform a genome-wide knockout screen using the CRISPR-Cas9 system. And in this system, we have our cells expressing NMD reporter and the Cas9 protein, and we're infecting these cells with a pool of lentiviruses that are targeting the all genes in the human genome. And in each, we have 6 guide RNAs targeting each gene. So in this system, a single cell gets a single guide RNA, and then we collect cells that have inhibited NMD, and these are the cells with high mCherry to GFP expression. And so after deep sequencing, we can now identify genes that cause the knockout of an NMD important of a gene important for NMD, which caused this inhibition of NMD. And what we found was among our top hits, a number of the genes were already known to be important for NMD, which was a nice validation of our reporter system and the CRISPR-Cas9 screening system. But importantly, we also found a few other genes that were not associated with NMD before. And some of these were members of a spliceosome system, or U2 spliceosomal system, SF3B1, SF3B5, and SF3A3. And what was interesting is that SF3B1 is commonly mutated in cancers, especially leukemia and myelodysplastic syndrome. And we wondered whether the mutations affected the role of this gene in NMD and whether inhibition of NMD would cause cell death in these cells. So we set out to do this. So we had cells that expressed this mutation, the SF3B1 mutation, and then we inhibited NMD either using the knockdown of an NMD factor or using an inhibitor of NMD. And what we found was that these cells were significantly more sensitive to NMD inhibition compared to the cells that had the wild-type gene. What was most fascinating that we found in this research was the deeper connection between the spliceosome and NMD, and particularly the role of SF3B1 and these other U2 spliceosome genes in NMD, which are commonly mutated in cancer. And we found that the mutations in these genes, SF3B1 and U2F1, made cells vulnerable to NMD inhibition, which provides an avenue for developing therapies to treat cancer cells harboring these spliceosome mutations. Because splicing and NMD are very closely related, it was difficult to untangle these two processes to really pinpoint the role of these spliceosomal proteins in NMD. What is next in this, in the field of NMD, is to better understand the mechanisms of NMD. And what we can do is, instead of relying on these models, substrates of NMD that have premature termination codons, we know there are other mRNAs that have other features that make them substrates of NMD. And by using those, we could potentially identify specifically how these spliceosome factors promote NMD and better understand how each individual mRNA is targeted for NMD and decayed to generally understand the mechanisms of NMD in human cells.